this? Yeah, okay, perfect. Well, yeah, thanks for the invitation to speak here. Very nice conference so far, very intense. A lot of nice talks already that we've been listening to. We are switching gears a little bit, I have to say, so actually moving away from tox and safety more towards PK, and also bringing you towards that mindset that the marriage of organ-on-chips or advanced in vitro cell-based assays with computational models is what we as a company at least think is the way to move forward. But first things first, that's very bright. So we are a German-based CRO, ASCII Labs. We work fully digital. We are a team of 20 people roughly, from physicists to software development. We have everything in there. And we basically do biosimulation to support our clients, which are mainly pharma, crop science, or chemical industries, in their overall decision-making process for go, no-go decisions. So they have a bunch of data, we analyze them and model that, and can tell them whether they can move forward or not with a compound. We also, as a small CRO and startup, strive for strategic partnerships, so that's why these conferences are so nice. And we are also part of a few research, national and international research grants. What we offer is basically, can be divided into 4 pillars. One is, again, modeling and simulation. So there's a bunch of data out there. We develop a model for this, and we see what comes out of this. We also do training and a workshop on the software that we use. And we do consultancy, which is basically, bespoke model development. And we also have a software that I will be touching on just in a second that we co-develop and basically distribute. Now, for the modeling and simulation part, if we zoom in more towards the systems pharmacology part, we can divide this into 3 main areas. One is PK, so we have heard a bit about this, but also QSP type of modeling, so where you really want to zoom in and understand on the cellular level how antibodies and antigens, for example, are interacting. So this is something that we can work on here. And my specialty, and this is what I've been working on over the past couple of years, is really developing those digital twins for organ-on-chips or any cell-based assay, and trying to get more out of the data. So the software that we use actually is under the umbrella of the Open Systems Pharmacology Suite, comprises 2 softwares. One is PKSim, mainly responsible for preclinical animal as well as patient or special population simulations for compounds and their distribution. And again, this QSP type of model, MOBI, is really if you want to zoom in an organ and understand on a more cellular level how your compounds interact. Okay, really rough and quick intro. Now focusing on our view on where we see that you can apply organ-on-chips in drug development. And again, this really resonates well with that context of use that we've been hearing a lot about. So depending on the question that you want to ask, obviously we just saw a nice presentation on a more high throughput screening type of model. So if you really want to know very early whether you have a potential lead or a compound that could be promising, then that is definitely an area where you could employ these advanced in vitro cell-based assays. On the other hand, reducing the throughput quite a lot, but therefore increasing the content and the biological complexity that you get out of this, is for single or a few integrated studies. Here as an example for PK. Then you can also move this towards more multi-organ studies where you look for pharmacodynamics, so effects or toxicity studies, where you really need that organ-organ crosstalk to elaborate on a certain drug response. This gets ever so more complex now by adding the immune system. We move away from simply having hepatocytes. Now, I think most of the liver chips contain Kupffer cells and stellate cells, but again, depends on the context of use, what you want to do with this. And last but not least, Uwe Marx mentioned this in his opening lecture already. There is this vision of a human body on chip. While that is still futuristic, I think we are not too far away from this. To really put a lot of organs together on a single integrated platform and really emulate a lot of the biology that is present in ourselves. So we as a company see basically 3 main strategic entry points of modeling and simulation on towards the organ-on-chip field. So one is actually pillar one is referring to scaling. So we understand the human physiology, and we basically have mathematical models and algorithms that help you to design and take that information to the lab bench. So as an example, if you develop a liver and a gut chip and you want to integrate that on a single chip together, it's really important to know how big you make the liver with respect to the gut, and if you want to add the kidney in there as well, what's the ratios of the surface areas and the media volumes to really then get back data out of this that you can use in clinical translation. The second pillar then focuses on in vitro and in silico modeling. So here this is really developing these, what we call digital twins of the organ on chip and the hardware system together. And the third part is then the translation, so really taking the data from those chips and employ them in computer models of the humans to then predict clinical outcomes. I will be talking about the last two of those pillars and provide you with an example so that it becomes a bit more clear how we want to work with this. So first one is the showcase where I will walk you through a paper where they actually used a liver chip to determine the metabolism of a compound, and then how that got predicted, translated to clinical kinetics, and whether that worked or not. So first of all, this is really just a super high level summary, stepwise guide, walk you through, so there's not a lot of mathematical equations that you have to digest. So first of all, as always, you just perform your bioexperiment. And in the case of the liver, if you have it, you can, for every donor, look at your different donor-specific drug kinetics. And this is something that is unfortunately not measured regularly at all in the scientific community, I have to admit, is the binding of the compound to the media, or the proteins inside the media, as well as the binding to the hardware. The plastics, the tubings, everything that sort of like potentially could reduce the fraction of free concentration of the compound, which is, as we all know, the only concentration value that drives metabolism or tox effects. So if we, from the very start, don't have a good guess what the actual concentration is that the cells are experienced to, the outcome we get out of those analyses are skewed. And then we store that internally in a format that we can then further process. So brings me already to the second step, more on the computation side of things. So we develop a computational model or a digital twin of the organ of chip at hand. Then you do your computational analysis. In this case, you determine or fit the individual clearance value for every donor. But you also take into account the platform and media binding. So by knowing how much concentration actually is available for the cells to metabolize, you simply get a better estimate of your clearance value. Now, the third step is actually in combining all of those. So first of all, you need a digital twin of your organ on chip, but then you also need a digital twin of the human body that allows you to describe the distribution of the compound within the human body. And if you then combine this with, so then you get your organ on chip specific data, so namely here the clearance and diffraction unbound. You integrate all of that in one big software, and then you simulate your clinical distribution. Yeah, okay, this is nice. So on the top right-hand side, you see the red curve, which is basically the prediction with the shaded area of our computational models. And we overlaid this with clinical data from I think a paper out of the 80s or 90s. And in this case, really the prediction worked quite well. So again, the workflow was do an experiment in the lab with one compound over a couple of donors and predict this without the use of animals. And we could clearly predict the clinically observed kinetics of that compound in a smaller patient population. Well, there was a nice example where it worked, but also from the same paper, we looked at a different drug, propranolol in this case, and we could show that actually the translation here would not have worked that well. So it was a nice pick to show that in the paper, but actually if you do this for other compounds, you realize that there's a translation gap. So it was the same workflow, data from the paper, we developed a digital twin and then predicted this, and as you can see here, the Cmax value is about 3 times off, but actually the AUC will be even more off because of the long tail. So the question is, can we do better? Is there anything that we can improve to predict the clinical clearance values better? Well, there is, and that's obviously one of the reasons why I'm here. But I first want to walk you through very high level why we actually need those digital twins for the organ chips that most of you here are developing. So quick recap of how you would normally go about getting clinical clearance values. You have your 2D sandwich, suspension hepatocytes models or whatnot. You put your compound in this, you analyze this, you predict the data, and then you realize there's an empirical scaling factor that is still lacking. So it's a constant underprediction. Now, the view in the community was very early on that by improving the biomodels, but still employing the same workflow afterwards, that you would simply have a better prediction of your clinical values. Now, a quote from the paper is actually that they also needed an empirical scaling factor of about the same size to correct for that systematic underprediction. So we believe actually that you not only need to advance your biomodels, but obviously also the computer models and then integrate the data to really be better in that prediction. And we see this across different publications with different numbers of compounds. They have been tested over different years and also in different in vitro systems. So there's a systematic, constant underprediction that is somehow empirical and no one really understands why. But how do they actually model the on-chip biology? So far, this is really done with like a single mathematical equation. You collapse all the biology that you have into a single model from which your clearance happens, and that's it. Now, what we think, we want to actually preserve all of the mechanistic information that is present in the chips and also include a lot of the information from the hardware itself. So surface areas, media volumes, cell numbers, and by disentangling sort of the place at which the clearance happens, in this case, from where you actually sample, this would allow, at least that's our belief, to really come up with clearance values that are simply better or closer to the observed values in humans. So long story short, to wrap this up, it's a bit of a busy slide, but take a moment to really look into this. So we took the data from all of those papers. We had our internal analyses and advanced biomapping that we employed and came up with that density graph. So here you see 2 different distributions. The one in blue or greenish is basically the observed clearance values and the ratio towards the clinic. So it's on average only 2-fold underpredicted, but with quite a range of variability. So some compounds are even further over or underpredicted to the clinically observed values, and that is really detrimental because PK obviously is what PD drives at the end, and if you don't assess your PK properly, you have no grasp on how well you actually can assess PD at the end. Now, with our approach, we can actually do 2 things. First of all, we move that whole distribution close to 1, so the ratio of our simulated values versus the one you see in the clinic is very well matched. But we can also reduce the variability, so we don't have any outliers that totally screw our distribution here. We also went a step further and wanted to employ this for spheroids. The first analysis was for an organ-on-chip system, and here we're actually very happy to work with AZ together on that. It's only one compound so far, so further investigations are ongoing. But here we developed a bespoke computational model, and it's, again, as you can see here, it's not a lot of equations and parameters in there, but really disentangling the clearance from the permeability seems to be the key step. And the state-of-the-art value, I think, it's about 200-fold underpredicted over the clinically observed value. And if you employ our approach, we still underpredict this by a factor of 2. But it's only a factor of 2 and not 200 anymore. So you really get a better grasp on what your PK potentially will be. Now, coming back to that very first example of propranolol, where the prediction looked really bad, and again, it's a really busy slide, but you basically see 3 different distributions. So the blue curve at the very bottom is the prediction of the clinical PK when you use the clinically observed clearance value. And we overlaid that with pinkish dots from the paper. So that's sort of like our ground truth where we want to compare our results to. Now, if you take the value from the standard or state-of-the-art approach in determining your clearance value, you get that high overprediction in red. And it gets even more prominent if you look at the AUC of that. Okay. At the AUC of that curve. It's about a factor of 6 higher. If you take the value from our approach, you actually have a kinetic that is very close to the one that you would normally observe in humans. And really the take-home message here is that it's the marriage of, it's the combination of digital twin and biodata that really shows the predictive validity of these approaches. So, yeah. And with this, I think I will quickly jump to the last slide to be on time. Again, so it's sort of like a turnkey approach demonstrating a predictive power in a particular context of use. I think this is really like the main take-home message here. And it is the negative of, well, if you want to fail, well, then do it fast and very early. So you need to know if your compound has the right PK, and then from that you can actually elaborate on safety and tox. In line with the FDA Modernization Act, this would also give you a competitive advantage, because if you don't need to use animal models, well, that's nice, but it also would save you a lot of cost. And if you can simply predict better your clinical PK, well, then that's an advantage. On top of that, using computational models is also a lot more sustainable than using animals. Yeah, and with that, I would like to thank you for your attention.
Thank you, Christian, again. And time for question.
Thank you very much. Really interesting presentation. I'm wondering, to what extent do MPS systems actually offer additional value versus, say, a human S9 incubation with the right cofactors to generate your initial data set for modeling? Oh, sorry. Oh, I didn't realize that you cannot hear in the back. Yeah, no. So I was just wondering, I mean, you're basing all of this modeling on data that you're generating with a liver MPS system. With human cells, but to what extent does this MPS system generate data that is better than a human S9 incubation with your drug? Because I mean, I'm all for, you should always go for the simplest model that you can, and cheaper necessarily.
Yeah.
So, yeah.
Fair enough, and I used to be a very pro organ on chip only guy, but that is changing now, because it's really important to realize what's the right system for the question at hand. Mm-hmm. And that's why I showed this 3D spheroid example. That seems to be the standard in pharma to assess your clinical clearance value first. So if there's a certain drug class where you can only use 3D spheroids and it works with the computational model, you have a better prediction, then go for it. Like there's sort of like no need to go more complex at that stage. If you already get the answer you're looking for.
My question was exactly the other way around. I mean, to what extent are spheroids actually better at generating the clearance data than a human S9 incubation? That was...
Yeah, okay. Well, we only use data so far that are available in the literature. So having a bit more access to data from clients or pharma partners would help to sort of like really showcase over 100 or 500 different compounds. 3D spheroids are better in this particular set of drugs versus another set, for example.
Yeah. Thank you.
Yeah. Very nice presentation. In your approach, in addition to developing more sophisticated models, do you have to get the client to measure some of the parameters they don't normally measure, like the surface area measurements or the binding parameters, or you're using literature data for that?
Yeah. Didn't mention that. The approach we take basically results in barely any more work than that is currently done. So it's the kinetic data that you take anyways. But in addition to this, intracellular concentrations would really help, but definitely the media binding and the platform binding. And that's 2 more measurements, n equals 3, that can't be too expensive. I mean, yeah.
Christian, thanks for mentioning the binding of parental compounds or metabolites to the plastics which will interfere with the readouts, right? I'm struggling with this problem. Some of the parental compounds up to 80% binding, and of course it differs between the metabolites and parental compounds due to the chemical structure. So it's really important issue, and thanks for mentioning.
Yeah, and I mean, I think there was no question, but
No,
like as a comment and follow-up is, I mean, PDMS is, you know, very the thing not to test drugs on, but we actually develop computational models also to account for that. You would need some more measurements, obviously, but it's definitely possible to use those systems that use that type of plastic and still do drug testing on them. You just need to know how much and when your compound is being absorbed and maybe released back. And if you know that, the computation model is a great complementary work where you can supplement this and sort of like pre-simulate your expected outcomes.
In the same direction, I have a question, actually. Very good that you mentioned that the free concentration of the compound of interest is sometimes very different than the nominal concentration that you wanted to use. But one parameter influencing this very much is the protein binding property of the actual compound. So, as you said, this is normally not measured in the in vitro assays. However, with the modeling, and you know the protein binding property, you could include this parameter into your models. Have you done this? At least, not the binding to the plastic, but the binding to the proteins, to the albumin, or the FCS or so.
Yeah, so there were a few studies where that actually was measured. So then we took this into account and sort of like normalized our the observed data to just show unbound fraction. But there are also some other computational models where you, based on the percentage of albumin in your media, you can like already have an estimate.
No, I mean from other studies, the protein binding properties are known. So you have this parameter available, whether it is 99% or only 80%, then you can maybe use this parameter to include it directly into the mathematical model.
Exactly. For that, you would need to know the media composition at hand.
Yeah, of course.
If that's available, then sure, you can calculate this. If not, then you need to measure it.
Okay, so thanks again to all the speakers of this session and to the audience. Thank you.